Namaste everyone, I am Dr. Amit here. I hope you all are doing well. Welcome back to my YouTube channel at the rate Dr. Amit Official, your place for simple and high impact medical learning for all subjects. So if you haven't subscribed yet, make sure to hit that subscribe button and stay connected for more such videos on every topics from every subject. So in our biochemistry series, in our last video we discussed about G6PD deficiency. In today's video we are going to focus on an important topic called urea cycle. It's one of the exam favorite topic and very high topic to get many MCQs in USML exam, NMCL exam, postgraduate entrance exam, and all the professional exams of MBBS. So I'll explain urea cycle in a simple, clear and exam oriented way so that you can tackle with all the MCQs and all the questions from urea cycle. So stay focused and let's get started on our today topic, urea cycle. So our today topic is urea cycle. Okay, so before understanding this steps of the urea cycle, it's better to learn some basics from the urea cycle. Okay, so let's talk about the urea cycle. Okay, so urea cycle, let's define urea cycle. Okay, so urea cycle is the process, okay, by which toxic ammonia is converted into urea for excretion from the kidney. Okay, so for this only urea cycle is getting occur in our body. Okay, just to remove the toxic ammonia from our body by converting it into urea. Now amino acid, okay, amino acid catabolism generates common metabolites pyruvate and acyl coenzyme A, okay, which serves as a metabolic fuel. But during this process, excess nitrogen is converted to urea and excreted by the kidney. Okay, so when amino acid metabolism occurs, okay, so whatever the extra nitrogen formed there, so it is converted into urea and excreted from the kidney. Now let's understand these two lines. Okay, so amino acid has two parts. Okay, how many parts? Two parts. One is carbon and one is amino group. So let's first focus on this carbon skeleton. So the carbon skeleton, okay, this form the pyruvate and acyl coenzyme A, okay, and we all know this pyruvate and acyl coenzyme A enter TCA cycle, and from TCA cycle it produces ATP, and the ATP is used as a fuel. Okay, but the second part is amino group, which consists of NH2, that is nitrogen part, and this get converted to ammonia. Okay, and we all know that ammonia is toxic to our brain. Okay, that's why it's very important to convert this ammonia to urea and then to excrete that urea from the kidney. Okay, so now we'll talk about why this cycle is important. Now let's see. Okay, why urea cycle is important, why it is important to occur urea cycle in our body, because we all know that ammonia is highly toxic to brain. Okay, that's why its excretion is very important. So if urea cycle don't occur in our body, then ammonia will accumulate in our body and it will cause toxic effect to our brain. Okay, now where does this occur actually? So liver is the primary site where urea cycle occur. Now we all know about location. Okay, so while talking about location, it occur in both mitochondria and cytosol. Okay, I think in the very first video, I think from the biochemistry series, we talked about which are cycles occur in both. Okay, so we had a mnemonic of HUG, okay, heme synthesis, then U for urea cycle and G for gluconeogenesis. Okay, so urea cycle also occur in both mitochondria and cytosol. Now to summarize all this in one line, okay, urea cycle detoxify ammonia by converting it to urea in the liver. Okay, so this is the basic of the urea cycle. Okay, now we'll talk about this cycle. Okay, this urea cycle. So we all know this urea cycle occur in both mitochondria and cytoplasm. Okay, so suppose this is mitochondria and this is cytoplasm. So what happened in actually in urea cycle? Okay, the carbon dioxide and water combine to form bicarbonate ion. Now the student wondered why this is forming. The urea cycle is there, so it must be start with ammonia. No, actually this ammonia alone is very toxic. Okay, so our body is very clever. Okay, so it try to form a stable compound. Okay, so when ammonia combine with this bicarbonate ion, it form a stable compound. That's why the carbon dioxide and water combine to form HCO3, and then it combine with the ammonia form in our body. Okay, now the actually steps start from here. Now this bicarbonate NH3 form carbamoyl phosphate with the help of enzyme carbamoyl phosphate synthetase 1. Remember this enzyme name. Okay, this is also in short form it is also called as CPS1. Okay, this is the actually the rate limiting enzyme in urea cycle. Okay, now carbamoyl phosphate formed in the mitochondria. Now what's happened is the ornithine. Okay, ornithine is transformed to citrulline. Okay, so over here ornithine transcarbamylase enzyme is used. Okay, and this carbamoyl phosphate also used here to form citrulline. Okay, now from citrulline we get arginosuccinate. Okay, in this process the enzyme used is arginosuccinate synthetase, and during this process we use 1 ATP. Okay, and over here also. Okay, while making carbamoyl phosphate we use 1 ATP. Okay, so we use 1 ATP over here also. Okay, sorry, 2 ATP over here. Okay, so total 3 ATP, 1 ATP over here, and 2 ATP over here. Okay, now over here we use aspartate. Okay, aspartate is used to form citrulline to arginosuccinate. Now this arginosuccinate converted to arginine, and over here the enzyme uses arginosuccinate lyase. Okay, and over here fumarate form as a byproduct. Now this arginine is converted to ornithine, and over here the enzyme uses arginase, and in this step, okay, while forming arginine to ornithine we get urea, and this urea get excreted from the kidney. Okay, so this is all the process. Okay, now this ornithine. Okay, this ornithine, so again ornithine is formed over here. Now this ornithine again regenerate, and the cycle will continue. Okay, always remember that again ornithine is forming over here, and again ornithine will keep the cycle moving. Okay, now after that how many ATP is used? So 2 ATP over here and 1 ATP over here, so total ATP is 3 ATP is used. Now which one is the rate limiting enzyme? So talking about the rate limiting enzyme, it's carbamoyl phosphate synthetase 1 while converting HCO3 and NH3 to carbamoyl phosphate. So this step is rate limiting step. Now talking about this cycle is activated by what? Then this cycle activated N-acetyl glutamate. Okay, now urea cycle and TCA cycle is linked. Okay, how? So over here we see that this fumarate is coming as a byproduct. Okay, so this fumarate linked this urea cycle with a TCA cycle or Krebs cycle. Okay, this fumarate. Okay, sometimes the examiner asked. Okay, now while talking about this urea, okay, formula, this is there is 2 nitrogen compound NH2 and here is carbon. So 1 nitrogen is coming from ammonia. Okay, so this ammonia is giving 1 nitrogen in the urea. So while when the urea is forming, so 1 nitrogen is coming from ammonia, then and this N is coming from aspartate. Okay, we all remember that while forming citrulline to arginosuccinate we are using aspartate over here, and this aspartate is providing the second nitrogen to urea. Okay, sometimes the examiner asks the MCQ that there are 2 nitrogen in urea, so which compounds are providing nitrogen? So one is ammonia and second one is aspartate. Okay, and this carbon is coming from carbon dioxide. Okay, so this is all about urea cycle. Okay, so if you like this, I hope you can solve all the questions from the urea cycle now. Okay, and we will talk about some important diseases related to urea cycle in the next video. Okay, so if you like this video, please share with your fellow medicos and with your friends, and please subscribe to my YouTube channel, and stay tuned, keep working hard, and we'll meet in next video. Thank you.